So if electricity is the movement of charged particles, we need to be able to have a way in our cells to have charged particles move. We're going to, how do things move? Gradients. We need to create a gradient so that ions can flow, and that is going to be electricity. So let's draw this. What I want to do here is draw the concentration gradients across the cell membrane. You may remember this. So if we've got, I don't know why this doesn't always work. We've got our cell membrane here, which is not beautiful. Let me actually try this with a different pen. Okay, here is our cell membrane. We've got the ICF and the ECF. And we need to have a difference in ion concentrations on the outside and the inside. Why do we need to have that? So that we can have ions flow and we can have electrical signals occur. So if you remember, we have high sodium outside of the cell. We have high potassium inside the cell. There's a couple other important ions that I will come back to. These are the two I want to focus on right now. High sodium outside, high potassium inside. And we're looking just across this membrane. Let's say that this is like a little section. Let's draw our neuron again. Here is our cell body. And we are just going to zoom in to a tiny portion. We find a different color here. Zoom into a little portion like right here, right? That's what we're looking at. And this will be important to remember that we're just looking right along the membrane. That's going to be important as we start going down the axon, which we're not yet. So high sodium outside, high potassium inside. That's something you just got to know. You got to know it. You've hopefully, not the first time you've heard it. But also, is there something that you know about that helps to do this, to maintain these gradients, right? High sodium outside, low sodium inside. That's a gradient, a difference in concentration inside the cell and outside of the cell. There's a concentration or a chemical gradient for sodium. How? Why? Because of my favorite protein. You guessed it. I think I'm going to have it pop up here. Maybe sodium potassium ATP pump, also known as the Na plus K plus, because that is the same thing. ATP pump. The words are important. It's a pump. It's actually somewhat redundant because a pump means it uses ATP. So you could say a sodium potassium pump, and that would be the same thing. A pump is an active process, like pumping water. And that's in opposition to something like a channel that does not use ATP. It's not an active process. So this pump is a protein that's actually embedded within the cell membrane, right? It's a transmembrane or integral protein. And we're not going to go through the mechanisms, but it is going to open on one side and then switch and open on the other side. That's going to allow 3 sodiums to pass from the outside to from the inside of the cell to the outside, that direction. And when it opens up in that direction, it's going to pick up 2 potassiums and bring them inside the cell. And when it does this, it uses ATP. So this process of shuttling sodium out and potassium in uses ATP. It converts ATP to ADP plus a phosphate group. So that requires energy, breaking of that phosphate bond, energy that was acquired from glycolysis and all of the mechanisms our cells use to produce ATP from the sugars we eat. Okay, so high sodium outside, We're bringing potassium inside. Can you see how the sodium-potassium pump establishes the concentration gradients across the membrane? I hope so. This protein is present in all cells, not just your neurons, and establishes these differences. These differences, this high outside, this high inside, are going to be critical for understanding the action potential, which is going to be the movement of these ions down their concentration gradients.